Hello and welcome to my new YouTube channel. Today we'll discuss single objective evolutionary algorithm, how we can use them in architecture, and we'll discuss also Galapagos and Silvereye plugins for Grasshopper. Let's get started. To understand how evolutionary algorithm works, first we have to go back to genetics. In genetics, the single basic unit is gene. Alleles are all types of genes possible. Genotypes are sets of genes, and genotypes create phenotypes, so physical traits like blonde hair or dark hair or blue eyes. And in architecture, in today's example, our genes will be points, single points, and alleles will be all the positions possible of those single points. Genotypes will be clouds of points, so all points that are creating our phenotype, our pavilion. In genetic algorithm, we use crossover. So we choose the best types around the population and we cross over genes around those best types to get even more population. And here we can see how genetic algorithm works. So this function looks for the maximum, and on the left we can see the first population spread all over the graph. On the right side we can see 10th population that's getting really close to the maximum. And the closer to the maximum function will be, the smaller step it will take. But keep in mind that if function have more than one local maximum or minimum, your genetic algorithm can stack on one of them, and it doesn't have to be the best one, so the smallest or the highest one. To use genetic evolutionary algorithm, first of all, we need to create parametric model of our pavilion. Let's get started. First, let's create base points. Then let's start creating top points of our bottom curve of the pavilion surface. We are using construct point. And also let's decide what range of positions those points will be moving during the optimization. So let's say we'd like to move this point to each direction, Y and X, and then let's say we'll move this point, if the 5 is base coordinate, let's move 1 feet bottom and top. So let's set range for 2. Okay, so now we can see that our point is moving in some kind of range of positions, and all those point positions will be tested by our evolutionary algorithm to find the best position to create the best phenotype. So let's keep moving and let's create the rest of the points. Okay, and I will arrange those points, and there we go. Let's upload those points, base points from Rhino to Grasshopper. Okay, cool. So we have all our points in Grasshopper right now, and we are going to start creating the bottom curve of our surface. We are using interpolate, and the important part is to connect those points to interpolate command in the right order. So if you are starting, for example, from this point here, the next point should be this one, this top one, or this top one, and you need to go in a circular order. So holding shift, connect each of 10 points to the interpolate. And then to close the curve, you need to connect boolean true or just 1 to periodic to close the curve. Okay, so now we have bottom curve of our pavilion surface. So let's start modeling whole surface and the oculus on the top of it. We'll use patch and let's connect bottom curve to curve node. And to control the height of the pavilion, let's create another control point. Okay, that's way too high. Once again, we need to readjust the range of point. So that was the faster method of creating sliders. Let's add an oculus to this pavilion. A radius of our oculus will be another gene, so we need to adjust the radius and adjust the range of the radius. Now we need to cut a hole in our base surface. That's it. That's end of our modeling. But let's optimize solar radiation. So let's think that this pavilion has some benches, and we need to shade those benches as well as possible. So let's model those benches. We'll use top surfaces of those two benches as geometry for our solar radiation algorithm. So let's talk about Ladybug. As you can see, I copied this Ladybug algorithm from my other Grasshopper file, and I'm not going to go through the whole Ladybug plugin today in this tutorial, but I will show you briefly what I have here. What you would like to change in this algorithm for your own purpose would be probably file path. You'll need to upload your own EPW file from your own location, and time of analysis. Here I set up summer equinox and just 1 hour afternoon. In the main node, remember that you will have to change north direction, and you will need to connect geometry and context to Ladybug node. Context is always thing that shades our model, and our model is the thing that we are analyzing with Ladybug. In our example, our geometry will be those benches and our context will be the pavilion. Let's turn it on and see how does it work. We can see control points and subdivision surfaces that Ladybug analyzed. We need to readjust the size of the grid for our geometry. The more subdivision we'll have, the more precise but also slower our analysis will be. So probably this will work well for us. Let's check now test points. So the points where the algorithm tests solar radiation. We would like to set them around the height of person sitting on the bench, and we can see that they are way too high above the one bench and under the other bench. To fix this, we need to go to Rhino, click on the surface and use flip command. Now we can see that test points are above both benches, so let's adjust height. It looks like everything works well. Now we can start using Galapagos. Let's double click, type Galapagos. It will show up here. First of all, we need to connect all the genes and fitness function to Galapagos plugin. Galapagos is a special type of plugin. You won't be able to connect cables from left to right. You will have to connect cables starting from Galapagos to your gene. All genes are connected. Let's now connect Galapagos with fitness function. I usually use number command as intermediate node between Galapagos and our. Now we can double click on Galapagos and see what can we set here. Let's go through the basic setup of Galapagos solver. We can look for minimum or maximum value of our function. We'll look for minimum solar radiation, so let's change fitness to minimize. Then we can limit length of our analysis in two ways. We can control time of our analysis, or we can control max stagnant. So after how many very similar population, the algorithm would stop solving another ones. Normally I would use max stagnant, and it means so after 50 very much the same populations it will stop working, but for the purpose of this video I will use runtime limit and I will set it for 5 minutes. Normally it will take couple hours. Population is amount of genotypes in one generation, the more the better, but for purpose of this recording I'll just set 15. Initial boost controls how diverse the first generation of genotypes is. We can leave maintain in breeding as it is. Let's go to solvers. Normally you would like to hard preview to accelerate your algorithm, but for the purpose of this recording I will leave it here to show how the pavilion will change over time. After you will save your Grasshopper file and Rhino file, you can start solver. Okay, so I stopped the solver after 19 population. The dark red line shows average value, and if this line look like this, so it's just going to the top, it shows that it kind of found the best solution and it won't rather evolve our function any better. So here, for example, we can move around other populations. Let's first start from the first one, and we can see here fitness function results on this left bottom corner. We can see that they are very different, and this is absolutely okay. As you seen on the graph at the beginning of the presentation, those fitness function values will be all over the place, and going to the end, for example, they are very similar. The population is very close to the local maximum. And here we can see all the different values of all the different genes and genotype. So each of those lines that you can see here, those zigzagging lines, is one genotype. And each of vertical axis is value of gene. So for example, a line connecting each of z axis shows what are the values of the genes in this particular genotype. The further we go with our optimization, we can see that this range gets narrower and narrower till the point that it doesn't really change at all. All those genotypes are pretty much the same. So going to the last generation, we can reinstate different option. By clicking reinstate, it will create a Rhino model based on those genes, so based on those position of points. We can change them to see how they look, to find the best solution for us. Usually you would like to get the smallest one of the last population, reinstate this. After clicking OK, you will lose all those data. So remember to reinstate the best solution, click OK. Now we can test Silvereye. Silvereye is additional plugin for Grasshopper, so you'll need to download it from the internet and install it. Let's repeat the procedure of connecting genes and fitness. It works exactly the same as with Galapagos, so you connect from Silvereye node to gene, not the other way. Let's connect fitness function. Now we can double click on the plugin and let's see basic setup for Silvereye. It doesn't have as nice graphic interface as Galapagos. It's much more simple. We can minimize or maximize objective. So let's minimize our solar radiation. We can set number of generations. So let's set 19. We can set velocity, so how much different will be each generation from the latest one. We can as well set limit of time. We can set swarm size, so how many genotypes will be in one generation. Let's adjust 15. Then we can use initial position of your settings, but most important, usually you would like to hide preview. I would leave it just for the purpose of the recording. Then we can click start to start optimization. We finished our evolutionary optimization with Silvereye. As we can see, final result is much better than Galapagos. Actually, like 1/3 less than Galapagos value. Also, it was fairly quick. We can reinstate different phenotypes to see how pavilion changed over time. So we started from something like this with very high solar radiation, and then we moved through the analysis through phenotypes like this to our best phenotype. Same with Galapagos. After you will close this plugin, you will lose those results, so remember to reinstate the solution you like, usually the best one, and save and close. Then you can bake it. there we go. As you can see, those are our pavilions. The left one is from Silvereye, the right one is optimized with Galapagos. As we can see, with Galapagos we got much lower radiation, so I would strongly advise using Silvereye. That's it for this tutorial. Thank you very much. If you enjoyed it, if I learned you something today, like this video, subscribe to my channel so you won't miss my another video about multi-objective optimization.